Okay, I'm going to talk about the genetic nomenclature used to describe Drosophila melanogaster. Drosophila is a diploid organism, meaning that every chromosome is present in two copies, one from mom, one from dad. We symbolize that when describing it for wild type, for example. Wild type we symbolize with plus, and so we have two copies of chromosome, let's say chromosome 2 for the moment. And so we have two copies, each represented by a plus, and we draw that as like a fraction, plus over plus. And the two copies, one above, one below, each represent a homologue. Now there's a gene on the second chromosome called cinnabar, and that's symbolized with C and N. And so if we had a homozygous animal which was mutant for cinnabar, we would write Cn over Cn. Note that we don't typically write Cn plus, for example, over Cn plus for wild type, which it technically is. Cn plus is the normal wild type version, but that would be sort of pointless. We can just write the regular old plus and not have to worry about that. But we also don't write Cn minus over Cn minus. That is also not incorrect, but it's just not the way we write it. If we bother to write Cn at all, then that tells us that there's something going on with Cn. It's mutant. Otherwise, we would just not bother to mention it. So Cn over Cn would be a homozygous cinnabar animal. Cinnabar is an eye color mutation, gives you sort of orangey red cinnabar colored eyes. What if we crossed these two organisms, or these two flies, together? What would we get out of that? We would get a heterozygote, of course. One copy from one parent would be Cn, the other copy from the other parent would be plus. And so now we'd have a heterozygous organism, or heterozygous fly, same difference. One copy of Cn, one copy of plus. And note that it doesn't matter. We could write it either Cn over plus or plus over Cn. These are equivalent notations. Doesn't matter which symbol we draw on the top, which symbol we draw on the bottom. Similarly, it doesn't matter in the way we draw it whether the female contribution from mom is on the bottom or the female contribution is on the top. Nor does it matter, conversely, whether dad's contribution is on the bottom or the top. This is, in this particular cross, I'm assuming that cinnabar came from dad, but it doesn't matter. Again, it doesn't matter which symbol is on top or bottom relative to either the genotypes or the genders of the parents or the two genotypes in question. One thing to note is this is all well and good if you're actually writing things by hand the way I'm doing here, but if you're typing, for example, this fraction method or this fraction notation is a little awkward. And so if typing, then we simply use a virgule. And so we would say Cn slash plus, which is exactly the same thing as plus over Cn. Again, the order doesn't matter. So the virgule is simply taking the place of that horizontal bar in the quote-unquote fraction above. Okay. Move on. There are 4 chromosomes in Drosophila, numbered imaginatively 1, 2, 3, and 4. Chromosome 1, well, let's see. Let's just describe wild type here. And so wild type is plus over plus for chromosome 1, plus over plus for chromosome 2, plus over plus for chromosome 3, and plus over plus for chromosome 4. Now, chromosome 4 in Drosophila is extremely small. It only contains about 80 genes, which is far less than 1% of all the genes present there. And so, basically, very rarely is there anything going on in the 4th chromosome that we care about. And so, as a result, we don't bother to write that. We typically only write the 3 chromosomes, chromosome 1, chromosome 2, chromosome 3. And chromosome 1 is also known as the sex chromosome. Chromosome 1 is the X and Y chromosome. So this particular wild-type animal that I described here is a female because she has 2 copies of an X chromosome, plus over plus. If I was describing a wild-type male, how would I do that? So I would have a plus, a wild-type X chromosome over something, semicolon, plus, 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 everything else is wild-type, fine. Now, I could, and the way I'm saying this tells you this is not what we do, I could simply write Y there. But that, as I said, is not what we do. The Y chromosome instead is symbolized simply by a slash. sort of descending down and to the left from that horizontal bar separating the X chromosomes in a female. So this on the left is the genotype for a wild-type female, and on the right is the genotype for a wild-type male. Okay. Now suppose I had some mutations. So let's say I have a mutant in a gene called forked, which is on the X chromosome, and it's homozygous, and forked is symbolized with a small f, and it affects the bristles. So the bristles are twisted and gnarly. And by the way, none of the mutations I'm talking about here are present in your flies. I guarantee nobody in the class has the forked mutation on their mutant. You may have messed up bristles, but I'm just telling you it's not forked. So at little f over little f, this animal is homozygous for forked, and so it would show the forked phenotype. It would have forked bristles. Second chromosome, let's say it has cinnabar I, so it's CN over CN, and there's a mutation on the third chromosome called curled, which gives curly wings, and that's symbolized with CU. So this animal here is really messed up. It's got squiggly bristles, it's got funny colored eyes, and it's got curly wings. By convention, lowercase letters, lowercase f, lowercase cn, symbolize recessive genes. So lowercase is recessive. And again, this is a convention in Drosophila, not universally. Uppercase, in contrast, not surprisingly, stands for dominant allele, or dominant allele of a particular gene. And so there's another mutation called tuft, for example. TFT, capital T, FT. And so this is a dominant allele. Now, it's present on the second chromosome. Now, suppose I had a fly that had, let's assume that our poor fly is not so terribly messed up with everything else. It's perfectly wild type for the first chromosome, wild type for the third chromosome, but tuft, like cinnabar, is on the second chromosome. How do we symbolize that? And so what we would do is simply to write the first chromosome is plus over plus. Let me erase cinnabar here. Or I'm cinnabar, tuft, whatever. Let me erase something. Plus over plus for the first chromosome. The second chromosome, the animal has tuft and cinnabar. And let's make it heterozygous. And then semicolon, plus over plus for the third chromosome. So note the way we use separators here. Semicolons are used to separate different homologs. We have the first chromosome pair separated by a semicolon from the second chromosome pair, oops, and then there's a semicolon buried under that, separated from the third chromosome pair. Within on a chromosome, within a homolog, we use a comma to separate different genes. So we've got the tuft gene and the cinnabar gene, both in the second chromosome. In this case, both are mutant because they're both shown. They're not just left out as pluses. Right? If they were both normal, then you'd have a wild-type second chromosome, and it'd be homozygous wild-type over wild-type. And so this particular fly has normal-colored eyes because the second chromosome, if we want to expand out to be a little more thorough, just for clarity here, this is not the way we write it, but for clarity, the wild-type has the tuft gene on it, but it's got the wild-type allele for that. And it's got the cinnabar gene on it, but it's got the wild-type allele for that. Again, since they're wild-type, we don't bother to write that. We just stick with the plus. But in this particular animal, it's got one defective copy of cinnabar and one wild-type copy of cinnabar. And since cinnabar is recessive, as symbolized by the lower case, we know that this animal looks wild-type. The wild-type dominates over the mutant allele in this case. It's exactly the opposite for tuft. It's also heterozygous for tuft. It's got the mutant allele on one chromosome. It's got the wild-type allele on the other chromosome, but because tuft is a dominant mutation, or this particular allele in the gene tuft is a dominant allele, this animal is heterozygous. It's going to have tufted bristles. Normal-colored eyes, but tufted bristles. Okay. And I think that's everything we need to talk about as far as I covered multiple genes, upper lowercase, commas separating genes on the same chromosome or homolog, and semicolons using to separate homologs, symbolizing both pairs. One last thing. We can, I don't recommend doing this right out of the gate, but I recommend doing it this more complete way that I've sort of scribbled out here, which is now all but impossible to read. But if I have, let me just do it here in the corner. Plus over plus, semicolon plus over plus, semicolon plus over plus. Not much of a plus there on the second chromosome. That's wild type. Another way to write exactly the same thing, I can just erase everything here, would be to simply write plus to symbolize the first chromosome, semicolon, plus to symbolize the second chromosome, plus to symbolize the third chromosome. That's the equivalent. The semicolon is separating the first chromosome from everything else right here, and it's like, well, I've only got one chromosome. You can't have just one chromosome. So the inference is that it's homozygous. They're both the same, and they're both whatever this is. So similarly, this mutant up here with the F, the CN, and the CU, you could write that simply as F, semicolon, CN, semicolon, CU. And that's sort of how we would do it when you're more familiar with the nomenclature. As I said, I don't recommend starting with it that way. And I think that's everything we need to know.